I conducted a study on the development of gene editing tools on the animal model, specifically the zebrafish model, and I conducted it with my mentor, Dr. Krishan Arsari from Dartmouth College. So first of all, what is gene editing? Gene editing is a form of biomolecular technology that employs techniques to alter and edit DNA. The field was first developed after the discovery of the two-stranded DNA in the mid-1950s and was eventually developed to further our understanding of molecular genetics and was vital in establishing the foundation of many fields, including biology, bioengineering, and medicine. And as scientists began to link the occurrence of malformations in the DNA to the development of genetic diseases, researchers and scientists became more determined to understand these associations and patterns to prevent these type of cases. Consequently, it created the field of gene therapy. And over 70 years later, it remains as one of the most rapidly growing fields in biology, with new techniques being developed from experimental testing to therapeutic uses. And it has limitless applications that go beyond bioengineering and medicine, such as agriculture. Employing tools such as gene editing has allowed plant breeders and farmers to have plants to have the most advantageous traits within the environment to gain the most profit. So for some prior background, prior to 1953, our understanding of DNA was very limited, resulting in a variety of hypotheses about the structure, function, and purpose of DNA. Yet in 1953, through the works of Rosalind Franklin, she was able to discover the double helix structure and was vital in establishing those fields. 2 decades later, renowned scientist Werner Arbeit discovered restriction enzymes when he was experimenting with bacterial stains and infections, and he was able to discover that restriction enzymes had the ability to delete unnecessary or foreign DNA that could pose as a threat. And then building up to 1971, Paul Berg was named as the first scientist to recombine DNA from multiple species, developing the cut and splice procedure, sealing it into one individual piece. And from the development of past research, the first gene editing technique was developed, named the zinc finger nucleus, or ZFN, becoming the highest recognition technology of the time and revolutionizing the field. And as a result, scientists were able to discover the exact causes of these formations for these mutations would eventually inspire the development of the most popular gene editing tools today, including the CRISPR and TALEN tool. So for our methods, especially because me and my mentor weren't able to meet physically, we used past research papers and publicly available data just to analyze a data that's been already conducted by experienced researchers. And then we also delved into the understanding of zebrafish as an animal model within many scientific models. So these are the specific gene editing techniques I will be discussing in the next few slides. The ZMNS, the TALEN, the CRISPR-Cas9 tool, the base editing, and prime editing. So first is the zinc finger nucleases, or ZFNS, and it was the first gene editing technique developed in 1985, and it was a marvel at the time with its grand success and its high recognition potential, allowing scientists to delete any unnecessary or foreign DNA. And it works to recognizing an interval or section of DNA to specify a DNA sequence to join or cut molecular bonds. So you can see in the image on the left, there are the scissors, and it's basically just cutting that specific section of the DNA, which is why you have the left and the right sections. The next one is a transcription activator like effector nucleases, or TALEN, and it was initially developed in 2010 and inspired with the zinc finger nucleases, and it became a leading technique in the field and employs transcription activator like nucleus effector nucleases amino acids to recognize individual nucleotides in order to increase their ability to target specific sections of the DNA. Despite its similarities to the ZFNS, it remains to be costly and highly time intensive. Yet its success cannot be shunned in the dark, as it was able to correct the COLA7A1 dysfunction that causes skin to be very vulnerable, making its individuals more prone to chronic diseases such as skin cancer. And then you have the CRISPR-Cas9 tool, and it is one of the first gene editing techniques developed that became the face of all modern gene editing. And it employs a protein to make an incision within the DNA to allow for an alternate piece of DNA to be attached to the original. And and it has allowed scientists to target specific sections without compromising other sections, emphasizing higher recognition. And as a result, it has allowed scientists and researchers to imitate malformations in the DNA within animal models to study and observe. So as you can see in the image on the left, there are the genetic scissors that cut the double strands and that allows the tracer RNA to be attached. And then you have base editing. And it is one of the most simplest gene editing techniques that involves the substitution, addition, or deletion of a nucleotide to change the base of the three nucleotide sequence. It was derived from the CRISPR tool as a form of editing without breaking the double strands. And the development of this technology was a large step in the field as it allowed scientists and researchers to gain a deeper understanding in genetic diseases, as the majority of them are a result of nucleotide malformation, and it provided the opportunity to study genetic diseases in animal models through mimicking those same malformations in the animal model's DNA without compromising those double strands. And you can see that through on the left, it was originally the sequence of GC here, but then through the base editing, they were able to implement a uracil instead of the cytosine, allowing the DNA replication or the repair. And lastly, we have prime editing, and it is one of the more recent gene editing techniques developed in 2019 that reflects a similar technique, base editing. However, there are still key differences between the two forms. Base editing focuses its advantages in being efficient and being used often within animal testing. However, it is vulnerable to impact surrounding DNA around the intended section. Yet, prime editing is more versatile and is immune to the bystander editing, ensuring that the surrounding DNA is preserved from accidental edits. So through using the prime editing guide, as shown on the right, it allows for reverse transcriptase to occur through a cut of the double strands. So gene editing is still a very controversial topic in biomedical research, but how have scientists gone around this? So to avoid the ethical implications of human testing, scientists were able to find a placeholder for experimental testing through animal models. And it can be defined as a non-human species used in medical research since it can mimic the symptoms of a process or disease that is found in humans. And they've been found to have an extraordinary genetic overlap with human genomes and can be employed to study a biological process or disease that allows researchers and scientists to advance their understanding and knowledge. And they became a vital aspect of many biomedical research studies to test processes and diseases that would have been deemed unethical with human testing. So what is the zebrafish and why is it important? So the zebrafish is a small tropical fish native to India and is found in small, shallow rivers and streams, yet its main popularity stems from its versatility in biology. Despite mice and a variety of other animal models existing, zebrafish have significant advantages as test models. And presented on the slide, you can see a variety of different common research models, but the zebrafish is especially important. So the zebrafish as a model. So especially within animal model studies becoming more expensive, zebrafish have becoming more popular as an animal model to test due to their low cost and low maintenance. Although the mice remains to be the majority choice in experimental research, there has been a significant increase in choosing the zebrafish over the last 2 decades, and they hold great advantages in comparison to mice. For example, in prenatal studies, their transparent embryos are extremely useful in observing any significant changes. In addition, their genetic diversity is more varied as compared to mice, allowing scientists to be able to study a variety of different research topics on different genetic traits and conditions. So here is an example of one of their many advantages with their high rate of reproduction and rapid development. And on the top, you can see that their max lifespan is around 4 to 5 years, that allows scientists to prioritize research instead of really looking at the zebrafish. And as well, you can see through the embryo of how fast it just grows, allowing an abundance for researchers to work with. And then with their transparent and external fertilizing embryos, as you can see on the images on the left, as well as the video on the right, it basically allows the scientists to implement anything into the embryo. So especially when you're studying conditions that affect children in the womb, it's extremely useful just for studying any disease and how it can impact the body. And then general advantages and molecular features of the zebrafish. So you can look at their genetic homology, their high output physiological homology and maintenance. So especially with their genetic homology, they are pretty high, which allows that overlap of genomes. And with a high output, allows it allows for scientists to study a lot, especially since they produce 300 to 500 eggs a week. And their physiological homology is pretty close. That allows, again, just to study the genetic overlap. And the maintenance is low, allowing the scientists to prioritize research. And with the neuroanatomy of the of studying the zebrafish, so the human has over 1 billion neurons in comparison to the zebrafish with the 0.05 grams, and it becomes much easier to study, especially with the significantly lower amount of neurons, as well as just maintaining that equity and balance between finance and just managing the research as well as just performing it. And for our conclusion, through me and my mentor study, we were able to develop and really advance our knowledge of the history and development of these techniques for its applications in the current day. And although there has been major steps in the field to make it safer and to a higher precision, there still remains space to improve and grow. And many gene editing techniques are still extremely time intensive and prone to surrounding DNA to be impacted. So through further research and development, scientists and researchers could definitely modify it to make it safer and ultra specific DNA without having that opportunity to damage surrounding DNA. And with the majority of gene editing techniques still being controversial due to ethical concerns, scientists and researchers are still continuing to stick with animal models with its accessibility and equity towards many expensive research studies, yet further development to ensure its safety. It could soon be performed more often in humans with its emphasis on safety again. And these were the resources that we used. And are there any questions?